So amazingly, an adult by midlife has made an estimated 10 to the 9 or 10 to the 10 specific antibodies. And you didn't get sick 10 to the 9 or 10 to the 10 times. So that's just the number of foreign molecules your body has very quietly in the background while you're living your life protected you against. One of the concerns parents sometimes have when they decided to vaccinate kids is the fact that some vaccines are multivalent. They will have 3 or 4 different pathogens that you get protection from in a single vaccine. So for example, measles, mumps, and rubella is one of them. And I think they actually put the chickenpox virus in that one too now. So just contrast, 4 different antigens in a vaccine to the 10 to the 9 or 10 to the 10 different antibodies you're going to make in your lifetime. And you can see that it's not a tax on your immune system to handle 4 different antigens at the same time. It does it brilliantly, and it does it in your entire life. Now, when we look at antibodies, there are different types of antibodies. So we refer to these as immunoglobulin classes and antibody isotypes. So there are IgG. This is the one that people are most familiar with. It's the one that's in highest abundance. It circulates in your blood along with IgM, IgE, and IgD. And it is it's the one that's primarily responsible for neutralizing antigens or pathogens that are present outside cells. IgM has kind of got a really interesting pentavalent structure, and this is actually produced just in the very beginning of an infection. And usually what happens is the B cells that make IgM we call it class switching. They'll switch to IgG after a certain period of time. But usually in a new infection, we see IgM predominate, and then as your immune system gets a handle on things, it switches to IgG. We think of IgG as giving you much longer lasting protection, whereas IgM is transient protection, although I will say that some vaccines only stimulate IgM, and they seem to be still pretty broadly used. So I have had students question that, and I think it's a legitimate question, but we still certainly make some vaccines that only stimulate an IgM response. IgA is important because it's the form of antibodies that are secreted in your mucosa. So here's this picture here showing IgA all the way through your GI tract. So it's going to be the antibody that's present that lines your GI tract. And a lot of organisms come through here. So IgA is important in your immune system differentiating between friend and foe, and whether or not an immune response is warranted or not, IgA is going to be an important part of that process. I always believed that IgD was found on the surface of B cells, and it was part of the initial activation of B cells. Upon further research today, it seems like they don't actually know the function of IgD, so that's pretty interesting. Clearly, you don't have to know it if the scientists don't know it. IgE is very important to know about because it's the one that's involved in allergy, and IgEs will bind to mast cells and stimulate type 1 hypersensitivity reactions, which you're familiar with in terms of having sort of seasonal allergies and sneezing, etc. We'll talk a little bit more about that when we talk about hypersensitivity reactions. When you think about antibodies, they do 5 main things, and you should know these 5 things. This should be something that's in your toolkit. One of the things antibodies do is agglutinate organisms. Do you see how this one antibody has 2 different binding sites because it's got these 2 variable regions? And it can actually bind to more than one bacteria and cause them to clump together. This makes it a lot easier for a phagocyte because the antibody is stuck these 2 together, and the phagocyte can destroy them both at the same time. Okay, so it's very efficient. It helps the process of phagocytosis and your innate immune cells removing these pathogens from your body. Another thing antibodies can do is they can coat pathogens and result in enhancing phagocytosis. So we talked about opsonization last time, and I'm going to go back to my tortilla chip. Tastes really good by itself, but if you dip it in guacamole, it's way more delicious. So here we have our phagocyte, and it's the ability for this phagocyte to destroy this bacteria is enhanced by the coating of the surface of the bacteria with antibodies. Another thing that happens with antibodies is called neutralization. So here's a virus. We know viruses, they have to bind to the surface of cells and trick them, right? The virus itself will have keys, and it's going to bind to a lock. So we have a receptor binding that would happen, and that would allow that virus to enter into the cell. But if this virus is coated with antibody, or if this bacteria is coated with antibody, we refer to that as neutralization, because that often is sufficient to prevent, in the case of a viral infection, the virus can never get into the cell. And so you end up ending up with an infection. You know, if we all had antibodies to that new coronavirus that's going around, we wouldn't have to worry about it destroying people's lungs because, you know, the antibodies would neutralize it before it ever got a chance to bind. Antibodies are also really good at binding to toxins. And so there are toxoid vaccines. So thinking of diphtheria, thinking about tetanus, you know, these vaccines actually, instead of binding to the bacteria, they actually bind the antibodies that are made when you get vaccinated, actually bind to the toxin. So if the toxin is taken out of commission, then it can't do all of those harmful things. You know, for example, with tetanus, we know that it disrupts your neurons. It's going to interfere with those inhibitory responses when your brain is communicating to your muscles. So by neutralizing this toxin, you prevent the toxin from having that effect on your cells. Two other things that antibodies can do, we said that there's an antibody pathway for stimulating complement. So if antibodies are bound to bacteria, that can trigger the complement cascade. And of course, we know that there are 3 features of complement, and one of those is the lysis of cells, cytolysis by the membrane attack complex. We also stimulate inflammation, and we know that complement proteins themselves can bind to bacteria and result in opsonization as well as antibodies. The last protective mechanism has got the biggest name. It's called antibody-dependent cell-mediated cytotoxicity. And we say ADCC is how this is typically referred to. In this particular case, antibodies are binding to parasites. So this is really large. Look how big this parasite is compared to your cells. So this is a really big problem with cells for your immune system. These large parasites that are bigger than your immune cells, they can't just phagocytize it. The parasite's much bigger than the cell. So when the antibodies bind to the surface of these parasites, eosinophils can then bind to this. Remember, we talked about the constant region. The eosinophil can bind to the constant region, and it will secrete lytic enzymes, molecules like perforin, to start destroying this particular pathogen. So again, we can't phagocytize this big pathogen, but we can certainly exert some chemical warfare on it. And then this picture here is just showing what that looks like. When you have a whole bunch of white blood cells attacking a nematode, you know, there's strength in numbers here. That nematode is way bigger than any single cell, but when you have an army of these cells, all producing nasty chemicals and binding to the surface of that nematode, in this particular case, it looks like the immune system is going to win. I have another picture of ADCC, a little bit more detail here. So here we have our antibodies binding to the surface of the worm, and that's going to trigger macrophages, also eosinophils. These are our innate immune cells, but here they are shaking hands with our adaptive immune system, producing lots of nasty chemicals and helping destroy the larger parasite. This is a parasitic fluke, and here we have eosinophils binding to the fluke, trying hard to destroy it so that it can't complete its life cycle and make its host ill. So that's ADCC, antibody-dependent cell-mediated cytotoxicity. And I think it's pretty interesting. It's definitely something we haven't talked about, which is, you know, how does the body handle large pathogens, things like worms, things like larger protozoa, et cetera. They have tools for handling cells that are as big or bigger than the immune cells that you have in your body. That doesn't mean they always win. Worms tend to be pretty hard for your immune system to clear, and they tend to cause chronic infection. But there is a system when it works. All right, this is just a cute cartoon here. We show these are bacteria and they're causing an infection, and this is just to reinforce that idea. Here's our B cell producing antibodies, and the antibody lands on this particular bacteria, and it's going to neutralize this bacteria so that it no longer can be part of that infectious process. So that's antibodies. It was quite a long detour. What we want to do now, though, is talk about B cells and T cells and their different roles in the immune system. And so we're going to start with B cells because B cells are where antibodies come from. Try to get a sense of how B cells are able to produce all these different antibodies that help protect us from infectious diseases that we've seen at some point in life.